So Dharshini spent 3.5 years with Jill Cornish and I. So for those of you who don't know, Jill Cornish runs the bone group over at the med school. And she spent that time trying to integrate a computational framework to look at anabolic bone treatments. And she set herself two challenges. Well, I set her two challenges. Two challenges. The first one was to try and integrate this framework into a real human clinical trial. And as far as we're concerned, no one had done this before. And the second one we tried to do was, and this was probably the real challenge, and it was kind of nerve-wracking at some points, to do it in a blinded manner. So a lot of people who do modeling, they kind of like to know the results before they do it, and then do a bit of tuning and customizing. Dharshini did everything blinded and did not know whether her models were the same as the micro CT and nanoimitation until after the key was broken at the very end. So we sat down and we do it. So that added another dimension, and it really put her models to the test. So I will let Dharshini tell you about the many adventures she had over those 3.5 years and what she's been doing since.
Thanks, Justin. Okay, so the title of my PhD was Integrating Micro-CT and Computational Bone Mechanics to Evaluate Bone Treatment Outcomes. Now, before I talk about any of this, I've got a quick video just to remind everyone the process by which bone remodels. So there you've got the surface of the bone, and in red is an osteoclast. Now, osteoclasts are responsible for breaking down bone or bone resorption. So there it goes, it burrows away at the bone. And once the osteoclasts have done their work, the osteoblasts, which are there in blue, they come in and they're responsible for building new bone or laying down new bone matrix. So in normal healthy individuals, we've got a nice balance between the osteoclasts and osteoblasts, but sometimes there are imbalances in the system and several conditions can occur, one of which is osteoporosis. So treatments for osteoporosis can be split into two groups. We've got anti-resorptive therapies and anabolic therapies. Now, anti-resorptive therapies are very common, and these include things like bisphosphonates. They target the osteoclast to stop bone breakdown or stop bone resorption. Now, anabolic therapies, not so common, and the only therapy that's actually available at the moment is parathyroid hormone. Now, unfortunately, parathyroid hormone is quite expensive and can only be prescribed for up to 2 years due to safety concerns. So there's a lot of interest in developing new anabolic therapies, mainly because their job is to build bone. And over at the medical school, like Justin said, we worked with Jill Cornish, and they also have a lot of interest in developing these therapies. Now, as we know, with any new therapy or drug, we need to carry out clinical trials, and they can be expensive, time-consuming, and you need large treatment groups. And so this is where we propose modelling comes in. Now, modelling is able to inform those clinical trials and minimise treatment groups. It's fast to solve, and you can run large sample sizes at no real extra cost. And you're able to correlate treatment outcomes to specific bone features. So putting all that together, we came up with a hypothesis. Modelling can reproduce standard clinical metrics in a double-blinded manner. Hopefully by the end I would have convinced most of you or all of you that my model can do that. So this is the outline for the talk and pretty much how I set up my thesis as well. Firstly, create a validated pipeline for converting 3 micro-CT into FE models. And then we applied that to 2 big studies. The first one, the influence of fluoride on bone strength, and that was a human clinical trial. And the second one, evaluate the influence of material properties derived from different scales and sources as part of a murine study into whey protein diet. And finally, we had a much smaller study looking at sham and OVX surgery in a mice model. We also applied the framework to 2 collaborative works, which I'll talk about at the end, and I'll just briefly also talk about what I'm doing now, and then I'll finish up. So this is the framework. We start off with volumes of interest of trabecular and cortical bone from micro-CT, which then get voxel meshed. We then printed out one of those bones from the fluoride study and carried out an Instron compression test, and from that derived a yield criteria for all the bones, which were then applied to the FE simulations, and then eventually evaluation of the influence of those anabolic treatments. So specifically, the SkyScan 1172 micro-CT scanner was used to scan the bones, and that's just around the corner. And for mice bones, we used an X-ray voltage of 50 kilovolts and a voxel size of 5 micrometers, all of which are standard. Like I said, our first study was a human study, and we had trabecular bone from that. And the second and third were murine studies, and we had both trabecular and cortical bone from those ones. So once the scanning was done, I then sat down and segmented out the volumes of interest for trabecular and cortical bone, resulting in models that look like that, and also obtained a number of standard micro-CT indices, bone mineral density for trabecular bone and tissue mineral density for cortical bone, and those are from micro-CT phantoms, and also standard indices for trabecular bone, percentage bone volume, trabecular thickness, separation and number, and for cortical bone, bone periosteal and endosteal volume. Those models were then voxel meshed using hexahedral elements in hypermesh, and we chose an element size of 10 micrometers for trabecular bone and 40 for cortical bone. We then carried out what we believe was a novel approach to validate the models. One of those biopsies was printed out in 3D and subjected to an Instron compression test until we achieved these little microfractures, which is what we circled here in blue. The exact same conditions were simulated in an FE model, and the model was able to predict 7 out of those 8 fracture sites. And so that was a yield criteria that was derived from that experiment, and that was what we used as the failure criteria for our experiments. So once we had that failure criteria, the actual bone samples were simulated, and the sample was said to have failed once that yield criteria was reached. Now, the failure of samples were assessed across the treatment groups, and the failure values were also correlated against standard micro-CT indices. Okay, so the first study we applied the framework to was the influence of fluoride on bone strength. Now, fluoride has been around for a while. It's just elemental fluoride, and it has been tested in a number of bone experiments because it's been shown to have anabolic actions on bone and stimulate the synthesis of the bone matrix. Now, early studies used fluoride in quite high doses, and those studies showed no real improvement in the cortical mineral density. Some showed an increase in the trabecular thickness, but no change in the mass of the connectivity. And many studies reported an impairment in the bone mineralization and an increase in the fracture rate. And also because the dose of fluoride was so high, several studies reported adverse effects such as nausea and vomiting. So more recent studies have used fluoride in much lower doses in slow-release formulations, and these studies have shown mixed results. While some have shown a reduction in the vertebral fracture, others showed no real change. So what we really needed was a current clinical trial. And that's what they did over at the medical school. Now, 394 women received information about the study, and 180 postmenopausal women with early signs of osteoporosis were chosen and randomized into 4 groups. And the groups were placebo, which was also the control group, or they were given 2.5, 5, or 10 milligrams of fluoride every day for a year. And at the end of that 1 year, 23 women volunteered biopsy samples. And so these were the sort of goals for the study. Evaluate the influence of fluoride on the mechanical strength of bone by modeling. We looked at different loading modes, compression, shear, and torsion loading. We wanted to see which micro-CT indices correlated with mechanical measures of strength. And finally, we had some 2D histomorphometric data, and we wanted to just to see how that compared to the 3D data. And before I go on, I just wanted to show these images of some of those biopsy samples that had been micro-CT'd. I think they're really beautiful images. I think everything that's been micro-CT is really beautiful. But if you don't agree with me, they're also quite informative because you can very clearly see the progression of bone from normal through to osteoporotic quite nicely. Okay, so these are the first set of results from the model. So this is the first time we put the model to the test. In blue, we've got normalized material properties, so we're looking at the influence of the architecture of the bone only, and in red, an estimate of material properties from DEXA BMD. We've got the treatment on the x-axis and the failure on the y-axis, and we've got compression, shear, and torsion. So if we look at normalized material properties, you see a concave trend with a minimum at 5 milligrams for each of those loading conditions. And with the estimate of material properties, it's just a declining trend across the groups. So like I said in the hypothesis, and like Justin mentioned earlier, all our studies were double-blinded in design. So up until this point, I didn't know what the outcomes from the clinical side were. So once all the modelling was done, we had a look at the total body BMD. We want to look at it at 12 months because that's when the biopsies were taken, and we saw the same trend, a concave trend with a minimum at 5 milligrams. And then we looked at percentage bone volume, which is a standard micro-CT parameter, and again, same trend, concave trend with a minimum at 5 milligrams. So while it was quite unfortunate that fluoride didn't have any significant effects on the bone itself, we were quite happy because our model was able to predict the same trends, and it was the first time we were able to test out the model. Now what we also did was we wanted to look at some correlations between those micro-CT indices and the failure force, and again in blue is the normalized material properties and in red is the estimate. And we showed that the normalized material properties for each of those loading conditions were much better correlated compared to the estimate. And that's percentage bone volume and bone surface density, and we've got trabecular thickness and intersection surface. And like I said earlier, we also had some 2D histomorphometric data. We wanted to see how well, or how that compared to the 3D data. And so we did some correlations, and we expected the 3D data to be much better correlated, and sure enough, it was. So to sum that up, this is one of the few times computational modelling was incorporated into an actual human clinical trial. Unfortunately, low-dose fluoride had no significant effect on the mechanical strength, and that was consistent with the bone density analysis. There were non-significant reductions in the mechanical strength, and that was consistent with previous studies. Normalized material properties, or the architecture of the bone, showed a double-blinded predicted concave trend, and that was consistent with percentage bone volume and BMD. And 2D histomorphometric data was not an appropriate substitute for 3D micro-CT. So once that study was all wrapped up, we moved on to the second study, the influence of a whey protein diet in a mice model. So in this study, we had 29 female 12-week-old mice randomized into 3 groups, placebo or the control, or they were given 0.1% or 1% whey protein. 2 things. The reason we looked at the whey protein is at the moment I'm looking at the influence of an anabolic called lactoferrin. Now lactoferrin is dairy derived, and so it's its carrier, beta-glycan. Now beta-glycan is the whey protein aspect of the study, and so before we went ahead with the actual anabolic, we wanted to look at whether or not the carrier was having any influence on the bone. And secondly, from that fluoride study, we only had an estimate of material properties, so we improved on it this time and looked at the influence of material properties from different sources on model predictions. So to do that, we teamed up with Chemicals and Materials Engineering and did a number of mechanical tests: 3-point bending, nano-indentation, micro-indentation. And this time, when I scanned the bones, we scanned micro-CT phantoms, and so from that we were able to get bone mineral density and tissue mineral density values. So this is how we approached it. Firstly, we wanted to look at the influence of the whey protein treatment. And then, can computational modelling predict trends within trabecular and cortical bones separately by assessing geometry alone? And finally, do the model predictions change when material properties are derived from different scales from different sources? So this is the results from the modelling for the trabecular bone. In blue, you've got average yield force, in red, peak yield force. Normalized material properties is the architecture of the bone. Then we've got bone mineral density, nano-indentation, and micro-indentation. If you look at the average yield force, you see a declining trend across the groups for each of those. And when you look at the peak yield force, it's the same trend except it's amplified, as you would expect. And only when material properties were derived from nano-indentation was there a significant difference between the groups. Cortical bone, it's all the same setup, except this time we include tissue mineral density and 3-point bending. The average yield force, you don't really see much of a change across the groups. Same with the peak yield force, except it's an amplified response. So again, that was all the modelling. Then we looked at the bone mineral density and tissue mineral density. So for trabecular bone, we showed a significant decrease across the groups. Now bone mineral density values themselves showed a significant decrease, and that was consistent with what the model was predicting. And for cortical bone, no real change across the groups, again consistent with model predictions. So we split that up a bit further just to see where those changes are coming from, and we saw that percentage bone volume and trabecular thickness showed the significant decreases across the group. There was a non-significant increase with the trabecular separation and a non-significant decrease with the number, which makes sense as those two have an inverse relationship. So with the cortical bone, the parameters are not as straightforward, or not defined as straightforward as the trabecular bone. So you can think of the cross-section of the cortical bone as a donut. So everything that's bone, so everything in blue there is the bone volume. Now, the periosteal volume is everything inside the periosteum, or the red circle, and the endosteal volume is everything inside the endosteum, or the green circle. Now, any changes in periosteal volume is indicative of bone deposition or gain, while any changes in the endosteal volume suggest bone loss or resorption. So then we looked at periosteal volume and endosteal volume. The periosteal volume, there was a non-significant increase. Endosteal volume, significant increase across the groups. But then the overall bone volume showed no real change across the group, and that was consistent with what the model was predicting. So to sum that up, as the whey protein levels increased, there was a consistent decline in strength in trabecular bone. Source of material properties are really important because only when they were derived from nanoindentation was there a significant decline in strength. In this case, the strength changes were only in the trabecular bone, and the trends were consistent with the micro-CT derived indices. Now, the last study we had wasn't an anabolic. It was looking at sham compared to OVX surgery in mice models. If you're not aware, OVX stands for overectomized, and these mice have had their uterus and ovaries removed. And as it says there, OVX mice are well-accepted models of osteoporosis, and it's been well documented that following OVX surgery, mice lose a significant amount of their trabecular bone. Now, both sham and OVX mice are commonly used controls in bone experimental work. Sham surgery just eliminates any biases due to the placebo effect, while the OVX mice actually have changes in their trabecular architecture, and that's attributed to a deficiency in estrogen. And so we had 22 female 12-week-old mice randomized into two groups, sham or OVX operated. One of the reasons why we looked at the study also was part of that lactoferrin study that I'm working on now. We had both the control groups, we had sham and we had OVX, and we wanted to see mechanically was there any difference between those two controls. So like I said, much smaller study, so the approach was quite simple. Analysis of sham and OVX operated mice from a modelling perspective. We wanted to see how BMD and TMD measures for trabecular and cortical bone compared to the failure force, and finally we wanted to look at the micro-CT indices. So what I've got there is a cross-section through one OVX and one sham femur, and just by looking at those images you can see that the sham femur has a lot more trabecular bone, a lot thicker struts. Just looking at it you can see that the sham just looks a lot healthier than the OVX. And sure enough, that's what you see with the bone mineral density analysis. There's a significant difference between the two. But when we looked at the average yield force, we showed no real change across the groups, but the peak yield force showed that trend, although it wasn't a significant change between the two. We then broke that down. Percentage bone volume and trabecular thickness showed significant differences, while the separation in number showed no real change between the two. Then we've got cortical bone, and those are the donuts I mentioned earlier. By looking at it, you don't really see much of a difference between the two. And sure enough, that's reflected in the tissue mineral density analysis, and again in the average and peak yield force. And there wasn't really much of a change in the periosteal, endosteal, or overall bone volume. So a couple of points. Now, cortical bone, no significant difference in computational strength or TMD due to bone architecture. With the trabecular bone, there were significant differences in the BMD, percentage bone volume, and trabecular thickness. The trend was only highlighted by the peak failure force in this study, and the trends were only in the trabecular bone, and that was consistent with our whey protein study. And just two things to note. Now, cortical bone, I think we all know, bears most of the strength in the whole bone, so there wouldn't have been much of a difference in strength in vivo. And also, like I said, bone resorbs endocortically, so any reductions in trabecular strength is just indicative of problems to come. So those were the three studies I did as part of my PhD. We also had two collaborations. One was with the University of Southampton, and they were looking at vitamin D deficiency in utero and how that affected the offspring in the future. So doses of vitamin D surpassing physiological levels have been shown to stimulate bone resorption. So they had these bones from very, very young mice, and these mice, they tested their bones by carrying out mechanical tests, but the bones were way too soft for any mechanical tests, so they brought it over to us, and we were able to apply the framework to it, and very nicely showed that the vitamin D deficient group failed at a significantly lower load compared to the control group. And later on, they had bones from much older mice in the study, and they very nicely also showed the same result. Then we teamed up with a medical school to look at a peptide called adiponectin. Now, adiponectin is secreted from adipocytes, and they have an inverse relationship with bone mineral density. So the study compared the wild type to an adiponectin knockout. Unfortunately, overall, there were no real differences in the bone strength or material properties. That is an oversimplification. There were some minor changes in the cortical bone when you looked at the micro-CT indices, but overall, no real differences. But what I thought was really nice is that this study just got published in Endocrinology this month. And like I said earlier, I think micro-CT images are beautiful, and the micro-CT images from that study is front page, so I like that. And finally, this is what I'm doing now. I have no results to show because I'm still in the analysis stage, but I'm looking at an anabolic called lactoferrin. Lactoferrin is an iron-binding glycoprotein, and it's found in epithelial secretions, particularly in breast milk. And it's been shown to have anabolic, differentiating, and anti-apoptotic effects on osteoblasts. So it's said to be a very potent anabolic. So I'm hoping I see some positive results soon. So just final 3 points. Now, applying the framework to a human clinical trial was a demonstration of modelling in practice. I had 3 separate studies that showed that modelling using that double-blinded design is able to predict similar trends to micro-CT measures. And then we also applied it to 2 external studies and that lactoferrin one now. And our model is able to isolate the effects due to bone architecture alone and evaluate anabolic treatments on bone strength independent of material properties. And so that's me. Over the years, I've had many people to thank for all their help. Justin, obviously, big help. Jill over in the bone group, my friends. But, but no, that's me done. Thanks.
Exactly on 30 minutes. So yeah, we have plenty of time, unfortunately, for questions. So where were those seated questions we previously had? So does anyone have any thoughts, questions, or queries?
So at the start, you said one of the motivations for using computational models is that you can run trials on much larger populations than human clinical trials.
Right, then?
The sample sizes here were sort of limited by the actual physical data you collect. So how do you think your framework could be extended to actually run larger than physical sample sizes?
So you're just saying, like, if I've got a really, really large sample size,
Well, so because I guess you're comparing it to normal clinical trials. Let's say as big as number of patients you can recruit. Does your method have that same limitation, or is there a way you could synthetically expand that sample size?
I don't get your question. Can you, like...
Maybe along the population
Yeah.
approach
Yeah, is that where you're going with this?
Yeah.
So I think you're saying if you could extract some of the key features of a model, for example, with trabecular bone, and then synthetically generate a larger population, and then run your mechanical test on them, that could potentially inform what the outcome would be in a very large cohort.
Group, yeah.
But you need enough to train it on region. So you've got like 30 to start with. Yeah, I think something along those lines. But it's also, since our models, I think, are matching like mechanical tests and things, we may not have to do all the nano indentation and 3-point bending tests and things that go on, which are quite time consuming and can be quite expensive.
You just do it on a subpopulation.
Yeah, you can do it virtually in a subpopulation.
Would you presumably still need micro-CT of the patient to get the trabecular structure? Because that was important, right?
Yeah.
I think so.
Yeah.
You wouldn't replace having to do bone biopsies.
There was a kind of a fortunate thing about that particular trial. We don't often get people donating biopsies. It's a painful process. But I think mean, this is beyond a little bit this PhD, but I've actually seen some work where people are actually starting to evaluate bone using MRI. And that's what I saw at the last ORS meeting anyway. And they're still comparing against micro-CT, but some of the later sequences are quite promising. So I imagine that would be a way of doing it, and you wouldn't have to worry about any ionizing radiation, I imagine. So it's something beyond, but of course Jill's group is about micro-CT and bone, but I mean, her framework is extendable. You just have to have the source of the imaging data. If it comes from a non-invasive source, all the better. Jeff?
So I think that's really, really neat stuff, Dharshini. As you know, I think the modeling is really cool, and I think you've demonstrated a lot of value in modeling and a lot of potential future application for that for the modeling space. But I'm actually thinking a little bit more on the clinical side. So you tried a few different treatments that were thought to be beneficial for bone, and you did a number of different tests to look at bone strength and resiliency. And found that in some cases there really wasn't any effect.
Yeah.
In other cases there was an effect, but only on some metric, but not another. So I guess in the big picture of your dissertation, for a clinical person, what would you say is maybe from your perspective the conclusion about these various treatments? You say like, oh well, if you're really trying to get whole bone health, you need to pair this with this in order to get trabecular strength and cortical, load bearing. Or can you make any sort of conclusions like that?
I think one of the main things that the model can do is it looks at sort of architectural influences. So you can, if say fluoride had some effect on the bone and it was just manifesting in the architecture, then that'd be a nice way to be able to test it using this model. So it's completely independent of its material influences. I think that's one of the main things about this model, that it can do that. Does that answer your question? No.
I guess it's just sort of intriguing to me, I guess, that some of the treatments that in the literature would be expected to be to show, demonstrably that it has a positive effect on bone strength, but you found pretty mixed results.
Yeah.
Would you say that that's actually really just consistent with the literature, that some of these have weak effects or maybe no effect at all?
I mean, in terms of the anabolic, the only one we really looked at with this one was fluoride. I think we were limited by sample size. We only had 23. Like you had 180 women, but only 23 volunteered biopsy samples. And the findings were consistent with the literature. It was really unfortunate because, elemental fluoride, that'd be a really nice way to treat osteoporosis. It'd be interesting to see what comes from the lactoferrin study, because that's in the literature. Everybody says that it's this really potent anabolic. But from what I've shown here is that I'm pretty confident in applying that framework to this lactoferrin study that we have now. So I think I'd be quite interested to see what comes from that. Yeah.
I can imagine that while a lot of the ones appear to have a no result, it was still good for the model, because the model was also showing the same thing.
Right.
But you saw the manifestation that things happen in trabecular bone quite often, but not cortical bone.
Yeah.
And that's a very common feature of a lot of treatments. They affect the trabecular bone and not the cortical bone. So your overall strength doesn't change that much because it's all pretty much borne by cortical bone.
Right, yeah. I think that's definitely a demonstrated strength of the model, is that it even predicted when things were maybe not as intuitive as, and I'm an outsider in the bone world, as I would have expected.
Yeah, I mean, some work that is ongoing with some guys at the School of Dentistry at Melbourne University. They work a lot with the Australian synchrotron, and so they get some very nice images. And they've shown that as you age, it's the trabecular bone that goes first, and then you get an endocortical expansion. So then the cortical bone starts to thin out. So you can maintain strength to quite an age, and then all of a sudden you may happen to be walking around and it just takes a little knock to the side of your hip and you get left with this thin shell and it fractures. And so the trabecular bone inside is actually mechanically playing a buttressing effect. Buttresses the shell that you have, and so that's what Dharshini highlighted, is that the loss of trabecular bone is actually a sign of things to come. You want to maintain that trabecular bone, and a lot of anabolic treatments are trying to stop that loss long before you get to the cortical bone loss.
So presumably that's actually where this where it should be going, because the unique feature of it is, isn't it, that the demonstration of the architecture
It's very important.
is novel, really novel by
Yeah.
the sound of things, which was really nice actually.
A lot of, as we were saying, when we were comparing with, so we worked a lot with Michelle Dickinson from Chemical Materials, and when they test things mechanically, confounded in the result is both geometry and material properties. They both play a role in the failure of that material, so they can't really isolate out the effects. Micro-CT does one good job of trying to highlight metrics associated with the geometry, but there's no good way of saying does this measure mean something mechanically. So her models was able to isolate out, and she could look at things with normalized material properties and say, I can tell you the architectural contribution to the strength is this, and then when you have the material properties, it's this, isolated out by looking at different sources, nano indentation, micro, 3-point bending. I think she found something very interesting with 3-point bending. Well, you found something quite interesting with the 3-point bending, how different they were to the others.
Yeah, so they ended up failing at a much lower load compared to nano indentation and micro indentation, and then we couldn't really explain it, and we ended up finding that in the literature that mice femurs shouldn't really be tested by 3-point bending. They twist a lot during the test, and it's just not a good source of material properties. Yeah.
For those people who are used to doing 3-point bending, if you study it in solid mechanics, it's based on an Euler-Bernoulli equation, and what people don't realize is you can't just apply it willy-nilly everywhere, because you're quite often violating all the assumptions of the Euler-Bernoulli. And when they do these short femurs, there's some twisting going on which actually contributes to the strength, but it's not accounted for in the Bernoulli equation. And so if you put it into the equation, your results, the answer you get is quite different from what you get if you put nano and micro indentation, which are actual estimates of the bone substrate. They're actual estimates of the material properties independent of geometry.
Yes,
So
and I guess the nice thing about what you've done is that it's relatively assumption independent. You're really using the information you have and saying, what does that do? And you can add to it with the knowledge of the material properties. So that's really nice.
Jeff?
So I have another sort of out there kind of question. So in the beginning, you sort of posed a model of bone regeneration and bone breakdown, right, with osteoclasts and osteoblasts. And so the treatment sort of focused on a blocker of osteoclast activity or an anabolic treatment for osteoblasts. But I was also thinking that surely there must be breakdown of bone that doesn't have to do with osteoclasts from entropy or even like mechanics, sort of micro fractures that occurs, just a physical, not a biological process. And so I guess I'm wondering, is much known about that? What, the sort of physical entropic breakdown of bone that doesn't have to do with osteoclasts? And if there's anything known, how significant is it? Because I'm just wondering if osteoblast treatments or anabolic treatment is more effective than osteoclast blockers, because an osteoclast blocker wouldn't prevent sort of microfractures, it wouldn't prevent, degeneration just from entropy, just from physical breakdown.
I suppose he's kind of mentioning that other modes of failure, like microfractures and cumulative damage, right?
Well,
are they accounted for?
Yeah, you can imagine a non-biological structure, like a British strut, right? That thing over time is going to fail just from entropy, right? Basically, and mechanical loading, right? And so there's non-biological things going on to a bone that's contributing to its failure. And fortunately, you have osteoblasts that can repair that. But so I guess I'm just wondering about that, if that might be maybe not a very big key, but just some component of the problem of bone fracture and bone failure.
I think in probably older people, that's probably a problem. Older people. Yeah, definitely.
But therein lies the difference between bone, which is this fabulous material which repairs itself against bridges and things which require you and I to go and fix them.
Right. We're the osteoblasts.
Maybe
We're the osteoblasts.
The thing is, and Tor would be very good to elaborate on this. When you move or you do exercise or anything strenuous, you're introducing micro damage.
Right.
It happens in bone and everything, and that's the reason why you get stronger. So the other major cell, which is the osteocyte, it's the mechanosensitive cell in bone which detects these mechanical microstrains and then recruits the osteoblasts and osteoclasts to do their work. The key thing is rest. So you allow a little bit of micro damage to occur and then you rest, and then it heals itself. And so that you get these building of laminar layers of bone, and they become stronger and stronger. So as long as I think the key thing is those other measures will only become important if you don't rest long enough. So I imagine high performance athletes who are, playing 5 days of cricket, and then they, constantly not resting. This is when you often get these injuries occurring because they want to quickly rush back. And I know some sporting codes have a rotation policy where, I know the All Blacks had it for a while, where even if you were fine and playing really well, they'd bring you off to let you have a rest period. And that basically kind of prevents a lot of these things from happening. So it's actually the rest and the allowing to repair which allows the osteoblasts and osteoclasts to remove that mechanism of damage. Plus, bone is a very nice structure. Consists of, if you look at the cortical bone, it consists of these osteons, little circular structures. And so I was speaking to Michelle Dickinson, who's quite interested in the fracture propagation. And if you try and fracture it, the fracture has to navigate its way around these structures. And in doing so, it loses most of its energy. It's all dissipated. It doesn't get all the way through. And so the actual architecture and nature of cortical bone helps to dissipate the energy. So quite often, unless it's a catastrophic fracture, lots of micro damage don't tend to have a major effect as long as you rest.
Right.
So yeah, I think that's key. So Dharshini's work is mainly just looking at the bone building, bone deposition, and bone resorption effects.
Yeah.
And it's a pity, I think, that a lot of them don't work. Apart from the one that's only clinically available right now, the PTH, parathyroid hormone. But it's quite painful. It can only be administered by injection. So it's something that people try to avoid. It has long-term effects as well. So the lactoferrin one is the one that we're kind of hoping for at the moment, because Dharshini has actually just finally completed all the micro-CT work, trabecular, cortical, and we've got all the nanomutation results. So she's about to walk across the bridge. Walk across the bridge to med school and get the key to break it, and then find out if we had a positive effect. So that's what she's about to do now. So hopefully there might be a nice significant effect there somewhere. But you did see her work with the vitamin D bones and things like that. That all produced a significant effect for an outside study, and so. That's again a nice feature of your modelling, where you cannot physically, mechanically compress the bones because they're too soft. You can do that virtually, and that's a cool thing too. So, any other questions? Any thoughts? Hey, you
I got a question. I guess probably more perfect for Dane is. Are there phantoms for the micro-CT through which you can quantify the material property?
Currently, there are the two phantoms they use for bone. I also have a set of aluminium foils, which I actually use, believe it or not, you can use for density for bone.
Okay.
It's the one area that I'd like to develop, we actually need standards and phantoms over a wide range. So if you're looking at a particular study, we'd have to look at getting the right standard, the right phantom, if you wish to do quantitative work. It's quite possible, but we just need the right phantoms. It's not cost effective for us to lay in a whole lot of phantoms, because they're not cheap.
Right. Yeah.
So I'd love you to do a study and then you can donate the phantom to me afterwards. How's that?
So what's the difference between all these different phantoms? Are they calibrated on different scales?
No, what it is, if you want to do quantitative work, you want a phantom or a standard that that is similar in terms of matrix and size as what you are trying to measure. Because, for example, trabecular bone is actually quite problematical because it's thin. And when it gets too thin, you start getting partial volume effects. So your density measurement is actually incorrect because it's not showing up the way you would think it is. So that's why the best type of phantom is something that is very similar to what you're trying to measure.
I guess that's why.
In size and density. So it's size and density.
Yeah, I guess that's why quite often you typically only get BMD for trabecular bone because it's
Yes.
an averaging,
Yes.
whereas you can get TMD, tissue mineral density,
Yes.
for the cortical. Although there are some studies out now, if you get it fine enough, you can still get TMD for trabecular.
Well, that's what the large group down at Christchurch using aluminium phantom, aluminium for exactly that, to quantify that and see what the errors are. But the biggest thing is if you know what your errors are, that helps. And you do need standards or phantoms in order to determine the errors. Yep.
Yes.
Hi. So I am a PhD student from Victoria University.
Yeah.
I'm actually working on composites, something that can be used in future as a bone graft sort of material. So I'm studying the mechanical properties both architecturally as well as compositionally. So I was just wondering if your model can be used to predict the mechanical properties of my structures. So they're 3D printed. They're based on a biopolymer and calcium carbonate composite. So my samples are really tiny. So studying the mechanical properties is very difficult. So can we use your modeling method to find out their mechanical properties?
Mechanical properties?
Yeah.
Yeah.
So, yeah.
You did one of the studies where you found out the fracture points were essentially similar when you did an Instron test.
Yeah, so that was our
My
validation
samples were
study.
not, are not as big to do any Instron test on.
Mm-hmm.
So very tiny samples. So I rely on DMA test to find out the mechanical properties, dynamic mechanical analysis, and probably do some more tests to find the Young's modulus and the fracture points and things like that.
Yeah.
So just wondering if.
Yeah.
Yeah, I mean that's the whole idea.
Yeah.
It's just a generic framework. Probably applies even easier to composite structures. And the idea is it's you're just loading it.
Yeah.
You can load it to failure, but you don't have to go all that way. You can still stay in the linear elastic region, and you can work out the homogenized material properties depending if it's an isotropic material, or you can have it in the transverse direction if it's transverse isotropic.
It's just that with my materials, they all don't have the same properties because the minerals in them are not distributed evenly.
Right.
So all of them tend to have different properties. So I don't know how the distribution affects their mechanical properties. So
if you have a spatially varying set of material properties, you can still lump it together and have homogenized material property for the entire structure that's made up of all these structures. But it's either going to be isotropic in the same direction, or you can basically find out what the average is in the axial direction and the transverse direction, and find out if you look at do you do mechanical engineering much?
No.
Okay, so if you look at a matrix of material properties, you'll have like Young's modulus in the axial direction, a Young's modulus in the other direction, and the other orthogonal direction, and the other terms will all be your shear terms.
Okay.
So that's what you do when you homogenize a structure. You're finding out what is its bulk material properties, how does it behave, which should match your experiment. So that's essentially what she had. She was estimating bulk parameters. It just so turns out that if you've got trabecular structures, there's a lot of pores in them.
Yeah.
So the bulk is taking into account a lot of those voids, whereas cortical bone, the bulk material properties are very, very close to actually what you nano-indent, because you've only got very small Haversian canals in them. You don't really pick them up depending on what scale you're indenting. So it's perfectly applicable to that kind of situation. It's just standard mechanics.
Could I bring her up to you maybe sometime tomorrow? She's here for the next 2 days.
Oh, she's here for 2 days only?
Yeah.
She's been here for 2 days already.
Yeah. Oh, okay.
We've been working, man.
Yeah. As we do. Yeah, sure. We can speak afterwards. By all means.
Thanks, Jeff.
Jude.
Has anyone taken these failure loads for sort of micro epi simulations and related them to, say, either muscle forces or functional loads and see, for example, what kind of exercise or what kind of fall would result in the kind of micro fractures that we're seeing here?
I'm not.
Not that we
No,
know of.
yeah.
No. I think it's a big jump to go from what you see at the micro scale all the way to the whole body loads. That's the whole idea of multi-scale modeling, I imagine. Tor would have an idea of what likely causes the bone to fail, but typically they're like catastrophic loads as opposed to a cumulative damage, which like generate these kind of micro fractures and then don't give them time to heal. But you can do it. I can see how you do it. You just put it into a multi-scale model and load it with muscles, take the stress at an integration point, move down another level, apply it, and then see if that load that's being applied matches the ones that we... The reason why she chose the phantom that she did was that its material properties are very similar to bone. When you run it
Yeah, plastic, yeah.
it was just very similar, so we thought, that's ideal.
That's exactly right.
So,
That's the way to do it.
yeah. Because we weren't allowed to fracture the actual bone specimens. They didn't allow us to.
I guess that's the great part about the model, is it doesn't destroy the sample. It can be
I think that was one of the
Yeah.
conclusions. We allow them to keep it for histology and other things that they do, and we just go off the CT. Looks like we have time for maybe one last question. Prolong the agony. The longest question I've ever
Just wrap it up, yeah.
All right then. I think we'll bring it to a close, and I do invite you all to come have a chat with Dharshini or myself, or talk amongst yourselves, and we'll have some small snacks here.